about carotenoid. We discussed this morning about carotenoid research and what carotenoids are doing, and we came up with two little questions at the beginning, so to say, as an icebreaker. I will show you here a few organisms. There's some plants and some fish and birds. Yes, and Mirika will now ask you a question. The first one is up to you.
Which are state animals / plants of California? A, the cardinal. B, the cardinal and the golden trout.
He's sharing his screen.
The golden trout, Garibaldi fish, and the golden poppy.
So what is right, A, B, or C? Anybody?
C, I think.
This is
Golden poppy. Golden poppy is California.
Yes, golden poppy. How about the Garibaldi fish?
It's a state fish.
The Garibaldi fish is a seawater state fish of California. And the golden trout is the sweetwater state fish of California. And the northern cardinal is certainly, is the screen lost? No, sorry. And the golden cardinal is certainly Ohio. Next question.
Johannes,
Yeah.
we don't can you share the screen?
Oh, yeah, it is on.
I think I'm sharing it.
Yeah, we can see.
Okay, it's good. Perfect. Yeah, looks good, Johannes.
Can you see now?
Yes.
Okay, the next question is now.
What do these animals / plants have in common? A, their names start with a G. B, their coloration depends on dietary
Carotenoids, yes, and
C, they are all acutely poisonous.
what is correct?
It has to be B.
It has to be B, yes, and actually there is B. We are already at the topic. They all display carotenoid as coloration, and that's the main reason why we all know carotenoids so well. So I will now start with talk, yes, and not longer talk about these beautiful animals, but go to the human eyes, yes. And also in the human eyes, carotenoids play an essential role for vision, yes. Beta-carotene, as you all know, is the metabolic precursor for compounds such as 11-cis-retinal, the chromophore of our visual pigments, and trans-retinoic acid, which plays an essential role in eye development. Here a few of the functions of beta-carotenes are listed, yes. But we do not only accumulate beta-carotene in the human eyes, and that's quite special. The human eyes, or more generally the primate eyes, you find similar things in birds, is also accumulating other carotenoids. In the human eye, in the macula lutea, the region where you have the highest density of photoreceptors, you can find high concentration of lutein, zeaxanthin, and mesozeaxanthin. And those carotenoids are thought to reduce chromatic aberration, increase visual acuity, and they may serve as scavengers of reactive oxygen species. In the last 25 years, my lab worked about the pathway which is depicted on this slide, yes. And I want to mention, yes, this pathway, talking about this pathways in this front of this audience is a little bit like carrying old to essence, yes, because many of those structures have been determined by Chris Balchevsky's group, certainly also Philip Kaiser and other people who worked with Chris. And I will only briefly go over some principles in this pathways. The first one is dietary carotenoids. They are dietary, have to be absorbed. This absorption is protein mediated for carotenoids and occurs by passive diffusion for retinoids. They have to be metabolized and then transported from the intestine where they are absorbed via the circulation in chylomicrons. Through the circulation, some of these carotenoids and retinoids are taken up by peripheral tissues. The larger majority ends in the liver where vitamin A is stored in large amounts, yes. And in phases when we do not have access to dietary carotenoids, the liver secretes vitamin A bound to the retinol binding protein, which is called RBP4. And then this vitamin A is distributed to peripheral tissues such as the eyes and further metabolized either to chromophore or retinoic acid. Today, I want to talk about a hub of carotenoid and retinoid metabolism. This is actually the outer blood retinal barrier. Here on the blood, as I mentioned before, Retinoids exist either bound to the retinol binding protein or in dietary chylomicrons, also probably in other lipoproteins. And these compounds, the retinoids, have to be really concentrated here. They have to be taken up in large amounts because they exist then in millimolar concentration in the photoreceptors of chromophore. The same is true for the carotenoids, at least for the human eyes, and they are transported either in chylomicrons or for beta-carotene in low-density lipoproteins or xanthophylls, such as the macular pigments in high-density lipoproteins. What is important for us is that this transport or failure on this transport, or more generally, problems at this barrier are associated with many ocular diseases, including diabetic retinopathy, retinitis pigmentosa, and the age-related macular degeneration. And therefore, it's, in my opinion, quite worthwhile to look into this and to understand this process in more detail. Today, I want to talk about two things which are happening at this barrier. One is the role of STRA6 in ocular vitamin A uptake homeostasis. And later on, I want to present and share with you some new results from my lab where we will discuss how carotenoids might be accumulated at this barrier. Let's start with the role of STRA6. The STRA6 gene was actually discovered by one of the major figures in vitamin A metabolism, basically in vitamin hormone function. This is Pierre Jambon's lab in Strasbourg, yes. And they were looking for genes which are retinoic acid inducible, and they found several genes and they named them simply by numbers. So stimulated by retinoic acid 6 was already shown that this must be a membrane protein, but after its discovery, it became quite quiet with this protein and not so much happened until Hui-san from UCLA very elegantly showed that STRA6 is the receptor for retinol binding protein. And this receptor was always postulated, but never molecularly identified. And thanks to the work of Hui, we now know that STRA6 is this protein, yes. And he also showed that RBP binds with high affinity to this receptor and mediates the uptake of vitamin A from RBP into cells. Filippo Mancia, and I think he already talked here on the seminar, and therefore I do not have to go into much detail here with his work. He then nicely showed the structure or dissolved the structure of this protein. He showed it's a dimer, a transmembrane protein which acts as a dimer. He characterized the binding site and also showed that STRA6 is complexed with calmodulin, which indicates a putative relationship between calcium signaling and vitamin A transport. We entered this field, or I entered this field, let's say it like this. I entered this together with Chris, yes, because both of our groups during that time, Chris already here at Case and me, myself, still in Freiburg, were working on STRA6, yes. And in Chris's lab, Marcin Golczak showed that several things, yes. And one of these things was that the transport of vitamin A, the uptake, is coupled to esterification via LRAD, and that LRAD drives this transport. That's quite important if you think about it, because this vitamin A has to accumulate in cells and has to be retained in cells. I will make a little bit of an analogy to glucose transport. Glucose has to be phosphorylated in cells, otherwise it would diffuse out, yes. And this would be also true for STRA6, yes, because STRA6, what Marcin also showed, and certainly together with Chris, that this transport is not unidirectional, it's bidirectional. STRA6 is a pore which works in both directions. What we were doing during that time, we looked for zebrafish, yes, and what we did, we knocked out the STRA6 function in zebrafish. There are two STRA6 genes in zebrafish, and we showed when we knocked out one variety of these genes that the eyes became vitamin A deficient. There were also developmental problems because of a wrong distribution of vitamin A. Some tissues suffered from vitamin A excess, other tissues such as the eye from deficiency. And we published this here in this paper together. This was, so to say, my big start with Chris, and we continued on this. What we then did, we were interested, where is STRA6 expressed in the mammal after we analyzed the zebrafish? And similar to the zebrafish, we observed it's mainly expressed in tissues which build a blood tissue barrier. One of those is the eyes, yes, and as shown here. Here in the eye, we have this high expression. Here we have an immunostaining. Hopefully you can see it because it's only this little, right? So, just a second, I'm fighting with the laser pointer. Here, this little red staining, that's the basolateral side of the RPE. We also have stained the RPE for RPE65 here, which is a marker protein, as you all know. And it's expressed highly at this barrier. Another place where it's highly expressed is at the brain, brain blood barrier, as here exemplified for the choroid plexus, and it's also expressed in the testis, as shown here, which also constitutes a barrier between the blood and the retina. We also generated STRA6 knockout mice, like many laboratories did. We did it by eliminating exon 2. Others did it by eliminating exons 4 to 7. The results were always the same. The mice could not express STRA6. But these mice were doing pretty well. They were born in Mendelian ratio. They were viable as long as they were bred on chow diet with sufficient vitamin A. So we became interested, what are the consequences? And this model was certainly, because it was viable, it allowed us to study the consequences now of STRA6 deficiency for the eyes. When we are now looking over lifetime to the vitamin A content in the eyes of mice which are deficient for STRA6, held on a diet here, which provides really ample of vitamin A. That's all these breeder diets here in the, what you have in the mouse houses, 15,000 international units. That's quite a lot. Think about, normally a recommendation for humans is about 4,000 units, 10,000 are already thought to cause adverse health effects, but mice are fed on the size. When you're now following the total retinoid content in the eye, you see it starts low, but then it recovers and it increases and it plateaus. Over here, this is our months. Over the months, yes. But when you compare it to wild type mice, here 1 month, here 8 months old, it's always less vitamin A. But it clearly shows you, yes, also without STRA6, the eye gets their vitamin A. And from this, we propose, yes, there is another way in, yes, and the other way in is most likely chylomicrons, the other lipid carrier of vitamin A in the circulation. And to demonstrate this, we did the following experiment. We took wild-type mice and STRA6 deficient mice, put them on vitamin A deficient and vitamin A sufficient diet for 12 weeks, and then we looked for the vitamin A content in the eyes, yes. And this is work which was first done by Mary Kelly in my lab and later on by Jean Moon. And what I will now present are a lot of data from Jean. She is working on this. And what you can see here are wild-type mice. They do not care whether they are for 12 weeks on a vitamin A deficient diet because they have those stores, yes. Those stores provide them with sufficient vitamin A so they can maintain vitamin A homeostasis. STRA6 mice already start with less vitamin A, but when you put them on a vitamin A deficient diet, they lose their vitamin A and really go severely vitamin A deficient. This tells us it's depending on chylomicrons. On the other hand, it nicely tells us that this is an excellent model, yes, this STRA6 mice, to study vitamin A deficiency in the eyes. And we did this, yes. What Jean did was she used this mice, yes, and she isolated the eye cups, yes. We proved this by measuring RPE65 expression and Rhodopsin expression. We got a really good enrichment. We isolated RNA, we did Illumina sequencing, and we mapped this to reference genomes. And what the result was the following, yes. Here's the heat map shown, and here's the principal component analysis shown. Here, vitamin A deficient. Here's a genotype, STRA6 wild type. Here, vitamin A sufficient, STRA6 and wild type again. And what you nicely see with this, first of all, these are all independent replicates. You see these mice were really reacting very, very consistently. But what you nicely see, there's one group which really stands out, and that's the group of the vitamin A deficient STRA6 mice, because they do not have vitamin A in the eyes, yes. And I will show you here now the volcano, but you see there's really a lot going on. Here we are comparing STRA6 deficient mice with wild-type mice on vitamin A deficient diet. remember, the wild-type mice can maintain their ocular vitamin A homeostasis. The STRA6 mice don't. You see all these changes, and when we are now mapping pathways, we basically find something what is quite interesting. We really see when the retinal pigment epithelium, also the choroid, becomes vitamin A deficient. We have downregulation of factors which are epithelial development, epidermis development, all these things. Basically what we observe here, and I will show you this in the histology in a moment, yes, it's really like this. When you are severely vitamin A deficient, you would expect photoreceptors are not doing well. That's true, I will show you in a moment. But what you also see is that the retina, the outer blood retinal barrier breaks down in vitamin A deficiency. You also see this with upregulation of immune response and stress responses in this epithelium in vitamin A deficiency. And here, this is what we already at the beginning observed, but we always found in this mice that you have this choroidal lesions, yes, you have these problems with CRP, and it's always basically like this, that it shows that severe vitamin A deficiency, likely through retinoic acid, we are working on this, we will share this data with you soon, yes, in a paper, is causing a breakdown of the outer retinal blood barrier. Let's have a look to these mice on vitamin A sufficient diet. Yes, and here the volcano plot is shown for STRA6 versus wild type on vitamin A sufficient diet. You see there's much less going on. And this is now mainly affecting vision. And I will now talk about what is going on here in this more or less low vitamin A, some sort of vitamin A deficiency, but there is still vitamin A likely, so retinoic acid dependent functions can be maintained. But what does this mean for the visual system? So this brings me back to the scheme, yes, and here to Ram, who did this analysis. We published this recently in Human Molecular Genetics, and we did a little bit more. I will show you at the end. And here again, the STRA6 mice, they start low. They are then increasing, plateauing, but never reach wild type level. What does this mean for photoreceptor, mainly rod and cone photoreceptors? And to do this, we looked at two different time points. We looked at 1 month, yes, where we have the severe vitamin A deficiency, and at 8 months when the system more or less recovered. Let's start with 1 month, yes. Here is rod. Here is rhodopsin expression. There is significantly lower rhodopsin expression in STRA6 mice. When you're looking for rhodopsin staining here with a rhodopsin with the ID4 antibody, you see it's nicely stained, but there is less rhodopsin, as you would expect. This is certainly also shown in ERG analysis. Yes, there is a significant and very pronounced difference between wild-type mice and STRA6 mice at this young age, likely because they do not have sufficient vitamin A to sustain rhodopsin synthesis and ligand or rhodopsin. And we have, I saw it, Vladimir Kevalov here in the audience, yes, and he is a specialist on this. He really can tell you the details. When you do not have unliganded rhodopsin, you always have it's basically like having constantly background light, and this background light really reduces the gain of phototransduction, yes. And to show this, that this is true for STRA6 mice, that they cannot accommodate the protein synthesis to chromophore synthesis, we involved Beata Jastrzewska from Case in this thing. And Beata is really an expert in rhodopsin, and she isolated for us rhodopsin from this mice and ran this spectrum. And what you can see here, the blue trace is the wild type mouse at 1 month. Here, STRA6 mice. So there's a 50% reduction of liganded opsin in the dark adapted stage. And this means that even in the dark adapted stage, and these mice were really nicely dark adapted, I can show you this, they have a lot of unliganded opsin, and this likely accounts for this problem what we saw in ERG, yes. And what we also did, this was Ramzan did 9-cis retinal supplementation to ligand the unliganded opsin. What you can see here in orange, the trace from wild type mice, age match, you see mainly 11 cis, yes, a little bit of wild trans, but no 9 cis. But here in this STRA6 mice, this is unliganded opsin, you see huge amounts of 9 cis retinal bound to this chromophore now, to the opsin now. Okay. Let's switch to 8 months. Yes, it becomes now more different, because what we saw here in this first slides was what we expected. We have too little vitamin A, too much opsin. This means unliganded opsin and problems with vision. But now we are looking at 8 months of age. Yes, and what we can see here at 8 months, yes, that this vitamin A content really recovered. Yes, and does it still cause problems? Yes. Does the mice now manage to have sufficient chromophore and synchronized opsin and chromophore synthesis? So results are shown here. Here we are looking first for the expression of the opsin. You can see it's still lower, but this was no longer significant. Yes. Here with staining for the wild type mice for rhodopsin, here for STRA6, there is obviously staining for outer segments. There is obviously not much difference. But when we are now looking to the ergs, we still have a significant difference between the wild type mice and the STRA6 deficient mice. They are still impaired scotopic responses. Here the A wave is shown. You would see the same in the B wave. We have published this, yes. And when we are now looking, thanks with the help of Beata, and we repeated this experiment several times, and did this very carefully, I think so. We always find there's 8% or 10%, depending on the experiment, of opsin, which is not liganted by the chromophore. And this may explain it. And certainly we also confirmed this result again. It's now significantly less. We quantified this, the data and the paper, yes. It's about 10%, yes, of the opsin remains unliganted, yes. Let's look to the cones, yes. This were the rods, yes. Let's look to the cones. And with the cones, it becomes more dramatic, yes. Let's first look to the green cones, to the mid-wavelength cone. Here's the expression at 1 month, here at 8 months, yes. Let's look at 1 month. We have lower expression of cone opsins. We basically cannot stain it, yes. Here these are sections. We do this also in whole mounts, yes. We have paper upcoming soon, yes, where we are doing flat mounts of the retina. You see the same. It's not because we selected sections where there is no expression. It's really, there is basically no opsin in those photoreceptors, no cone opsin, yes. And after 8 months, so vitamin A really recovered, yes, there is still very, very low cone opsin. It's the same or even more dramatically seen for the short wavelength cone opsins, yes. They are even not really expressed, yes. And when you are doing immunostaining, and I mentioned it again, yes, we did this also in flat mounts, we basically do not find immunostaining. So we looked for the ergs, yes, for the photopic ergs, yes, at 1 and 8 months. And what you can see here, they have very, very, very low responses of cone photoreceptors at 1 month. That's not so surprising. There's only little vitamin A. And at 8 months, when there's more vitamin A, it remains the same. There is basically no cone vision. We stained also for hemagglutinin to demonstrate that cone cells are still present. I saw people in the audience. People did this when he was in my lab. And what we could find, it looked like that cones survived. It was not like this, that they were simply dying out. Yes, it was so there were still cones, yes, but they did not get chromophore. Let's summarize. What can we learn from this? Yes, from why do we need STRA6? Yes, and I try to summarize this in this cartoon. Here we have the wild types, yes, and we have an ocular retinoid pool, yes. And this ocular retinoid pool is injected either by holo RBP4, yes, vitamin A, which comes mainly from the liver, and by chylomicrons, yes. And this vitamin A is then used to synthesize chromophore, which is depicted here. And this chromophore is then in mice delivered to rod photoreceptors or cone photoreceptors. And then it has to be recycled after lipase. But what does it mean? This looks relatively simple, and it's also relatively simple. But from our analysis in these mice, we can make a few conclusions. One of these conclusions is that the total retinoid pool, ocular retinoid pool here, yes, here expressed as a concentration, must be higher at all stages than the lysine acceptor sites of the opsins, yes. And only under this condition, now you can go to the STRA6 mice in a moment, we have in the dark adapted eye all rhodopsin and also cone opsins liganted by chromophore. We can also conclude from this that the total, or we see this in these mice, as the total retinoid concentration is homeostatic. Yes, it increases also in wild type mice over time, but then also it plateaus. Yes, it's not getting more and getting less, and this may be related to this in and outward transport by STRA6. Let's go to the STRA6 mice, because this is relatively boring, because everything is in homeostasis. What is happening when we destroy this homeostasis? By knocking out the STRA6. We have the following function. We have a reduced ocular retinoid pool. And the first thing what we can clearly show, and I think I could convincingly demonstrate this with the previous slide, the total retinoid pool is smaller than the acceptor sites in rod and cone visual opsins for the chromophore, yes. And this means, because this number is lower than this number, we find unliganded rhodopsins and immature cone opsins. What is very interesting, I didn't show you this too much, but I can share with you each their diagrams at wish. We always observed in this dark adapted mice, STRA6 mice, we never found retinal esters. There is no retinal ester pool in this mice, which is high enough, obviously, to sustain vision, yes. It's also interesting that retinoid plateaus, yes, you would basically, normally you would expect, yes, because when you have a constant, they are really living, these mice are living on a diet which is providing really ample amounts of retinoids. This is a lot of retinoids, but so there's constantly retinoids coming obviously in, yes. It's always too little and it's plateauing, it's not increasing. And this indicated for us there must be something like, there must be also losses of retinoids, yes. And one of the things what we ask us then is, are there losses of retinoids in the mouse eyes which have to be compensated by STRA6? And what kind of losses might this be? Is this bisretinoid formation or something else? Is there something going on? Is this process here, it's the outer blood retinal barrier, is this far more dynamic than we thought before? Is there a constant in and out? Is there a turnover of retinoids? And to answer this question, we thought, again, yes, the STRA6 mice might be a very nice model, but we had to do something else, yes, because we wanted to speed this process up. So we used albino background, yes. We used this black 6 mice, right? So black 6 mice with the mutation in tyrosine metabolism, which cannot synthesize melanin. It has been shown, yes, by many before, yes, that in the absence of melanin, light entering the eyes is substantially increased, yes. This is more light stress, as indicated here. I will show you in a moment that there is a light-dependent formation of aberrant retinoids, such as 13 cis and 9 cis. And it was also shown by Janet Sparrow's group at Columbia that there is a photolytic degradation of bisretinoids in albino mice, yes, which is not very healthy, by the way. So what we did was we now took our black STRA6 mice on a black 6 background, crossed them with these. We had CF1 generation, and we generated albino STRA6, black STRA6, albino wild-type controls, and black wild-type controls. And we now put these mice simply, we raised them simply in the mouse house. Ram did this, yes, and we looked under ambient light conditions, and we looked at these mice. And here we have the ocular retinoids, here albino wild-type, here black wild-type. There's not much of a difference, yes. When we are now looking to STRA6 mice, this is approximately 3 months of age, yes, you see black STRA6 mice, as before, have less retinoids. But when we are now looking to albino STRA6 mice, we have even more. We have really a loss of retinoids. They have significantly lower levels of retinoids than their pigmented siblings. Yes. When we are now going for rhodopsin, this was done again thanks to Beata. Yes, we are looking now for liganded and unliganded rhodopsin. Here, black wild type, albino wild type. Here, there is no difference. They can maintain their ocular vitamin A. All the rhodopsin is liganded. When you're looking for black 6, for pigmented STRA6 mice, you see it's significantly reduced. We are again in this range of 20–30% of unliganded rhodopsin. But when we are now looking for the albino STRA6 mice, there is a significant reduction of chromophore in the eyes, and therefore we have significant higher amounts of unliganded rhodopsin. Here is the isolated rhodopsin shown, yes. The next thing what we did was we put these mice either to darkness or to light, or let's bring this first, yes. We certainly did this control because we thought, look, it could be for some reasons or for whatever reason, these albino STRA6 mice have a different expression of rhodopsin or visual cycle genes or phototransduction genes. And it is here summarized here. This was not the case. This was not the reason why we see these differences. So we did the further experiment. What we now did, we either raised them under ambient light condition, dark light cycle for 4 weeks, or we put them to darkness for 4 weeks before they were raised under ambient light conditions. And here the results are shown. Here, the wild type mice, there is no difference whether they are dark adapted for a long time or whether they are raised. The mice then can maintain their rhodopsin. There might be a little bit, there's a tendency that this is less, yes, but it's under light conditions, yes, but it's didn't prove to be significant. This might be also the number of mice. If we increase this, we may see some significance here, but it's not much, yes. But when you're now going here to this STRA6 mice, yes, you really see now, yes, here dark raised albino mice, yes, you have significantly more chromophore than in light rays, yes. And this clearly demonstrates us, in my opinion, demonstrates that there is a light-dependent turnover of retinoids, yes. And if you do not have STRA6, like in wild-type mice, you cannot replenish this pool. This is, in my opinion, a relatively new function of STRA6, which was yet not so much considered. You need STRA6 to maintain and replenish retinoids in the eyes. We certainly also looked, what is the fate, or we are starting to look, what is the fate of those retinoids in this albino STRA6 mice? And here are some retinoid analysis. What we did here, this is the HPLC, these are simply mice taken from the mouse eye. Here we have pigmented STRA6 mice. You see retinal esters, they are not dark adapted. They are retinal esters, 11 cis, all trans, yes, and syn and anti. When we are now looking to this albino STRA6 mice under the same condition, you find a lot of weird retinoid stereoisomers, diastereoisomers, such as 9 cis retinal, but also 13 cis retinal. And we also looked, we sent this we sent this eyes to Janet Sparrow to Columbia, and she looked for us for bis retinoids. As what she found was basically, and this is confirming what she was postulating, showing before. When you have albino mice, you do not have any bis retinoid accumulation, yes, especially with the STRA6. STRA6 anyway have reduced bis retinoid like you would expect with less retinoids. But when you have them on an albino background in light and compared to dark, you even have less bis retinoids because there is a rapid photolytic degradation. And what we think, what we provided evidence here is that in albino background, that there is a rapid turnover of retinoids. That's probably not so new, but we show that you need STRA6 really under this condition to constantly replenish your retinoid pool. Let me summarize this first part of the talk. I think I'm relatively good in time. Let me summarize this first part of the talk. What I want to show you is that STRA6 protects the eyes against fluctuation in dietary vitamin A supply. I showed you this. When you put this mice on vitamin A deficient diets, they rapidly lose their retinoids. And what we found in our genomic analysis, and I think that's quite interesting, is that severe ocular vitamin A efficiency doesn't only affect the photoreceptors, it really also affects the integrity of the outer blood-retinal barrier and leads to a breakdown of the structure. What I also showed you, yes, I think so, is that STRA6 is required to adjust the stoichiometry between chromophore and opsins, and is critical for cone and rod vision, yes, especially also for cone vision. And this tells you, yes, somehow you need STRA6 to have a certain level of retinoids in the eyes, yes. And this also indicates, yes, I mentioned this at the very beginning. We are working and we are trying to figure this out a little bit. Others are probably doing this too. There might be something, there might be some crosstalk between STRA6 and the retinoid pool, yes. And it's interesting that it's and somehow influenced by calcium signaling. And in the future, we really want to figure out how this is regulated. How is this opening and closing of STRA6 regulated to maintain this retinoid pool on a certain level, yes, and distributes these retinoids elegantly into the pathways for cone and rod visual pigment synthesis without causing any deficiency in one of the paths, as we find in STRA6 deficient mice. Finally, with this analysis of the pigmented and albino STRA6 mice, I think we demonstrate that that's this process, yes, this transport of retinoids, that the blood retinal barrier is far more dynamic than thought. That there is, for example, a turnover of retinoids, which requires a constant replenishment of retinoids in the eye to maintain this pool. This process is certainly impaired in STRA6 mice and is no longer possible. Finally, we also show, yes, with this different backgrounds, yes, that pigmentation, yes, or genetic background certainly plays a role in ocular vitamin A metabolism and is influenced by, and that this is influenced by pigmentation and likely also other factors. This was the first part. I will now switch, make a really shift gears, yes, and change now from retinoids to carotenoids, yes. And this brings me back to the slide where we show the outer blood retinal barrier. I want to remind you that carotenoids are transported, dietary carotenoids in chylomicrons, beta-carotene in low-density lipoproteins, and xanthophylls, which are the precursors of our macular pigments in high-density lipoproteins. What you see here, this is the primate retina, yes, and here from Snorley's work, yes, a very early picture, yes, you have here the retina, here's the fovea of the retina. You see this yellow, nicely in yellow color. These are the macular pigments, which are really there in concentrations, which are so high that you can see them with the naked eyes, yes. And this is also was recently very, very nicely shown by Paul Bernstein's group at University of Utah. And what he was showing here, he developed a very elegant method, random spectroscopy, confocal random spectroscopy, to detect those carotenoids in donor eyes. And here is a picture shown. This is a beautiful article in PNAS. And you can see here that carotenoids accumulate in the fovea, and that's a major carotenoid which is accumulating in the fovea is zeaxanthin and mesozeaxanthin. Lutein is not so much accumulated in the fovea. Okay, I want to now talk about how carotenoids are enriched and how they are transported over this barrier, yes. And for this, I have to introduce a few players in this pathway. The first player I want to introduce is not Linda Thomas, but beta-carotene oxygenase 2, yes. And this protein we discovered almost 20 years ago, yes. And beta-carotene oxygenase 2, yes, we found it as the searcher for the vitamin A forming enzyme. That's not an enzyme which is forming vitamin A. It's an enzyme which is degrading carotenoids, yes, into smaller entities in beta ionones and the central C14 aldehyde by cleaving at position 9 and 10, yes. And I mention here Linda because Linda in her PhD work, she nicely showed, and this was not, it was quite difficult to show, yes, that human BCO2 enzyme is an active enzyme and also nicely showed how human BCO2 is transported to the mitochondria, because those enzymes exist, this enzyme exists at the inner membrane of mitochondria. I think hopefully you can see it, you see this little black dots. That's actually work from Chris's son, Greg Palczewski. He did this in my lab several years ago, and he nicely showed, yes, how carotenoids are entering the mitochondria, that carotenoids really are accumulated in mitochondria, that they're accumulating at the inner membrane of the mitochondria, and that the carotenoid cleavage enzyme is located there, yes. And we also developed knockout mice for this enzyme, yes, and with this knockout, this nicely shows that this enzyme has a very broad substrate specificity. It's a carotenoid scavenger. When you feed them zeaxanthin, they become yellow as corn. When you are feeding them lutein, they become orange like marigold flowers. And when you are feeding them astaxanthin, they become red like salmon, yes, because they cannot longer convert this enzyme. Genetic polymorphism in this enzyme are also found in nature, yes, and they are really responsible for some of those colors in nature. We showed recently together with a group in Colombia that a mutation in the carotenoid oxygenase influences the colors of domestic betta fishes, yes, and they account for this very red. And also, it's not only accounting for the overall color, it's also accounting for the color patterns, yes. And this is also very nicely shown here in this tropical bird with a collaboration with Michael Brown from the Smithsonian. This is still not published, but we hopefully publish it soon. And what he nicely showed that there are two varieties of this mannequins. There's little tropical birds living here in Central America. And there is yellow and white varieties. And there is a zone in Central America. It's in Panama, yes. And there's a zone where those two species are overlapping, yes. And in this zone, yes, he could identify that the BCO2 gene is one of the traits which is selective for this, because the females prefer the yellow ones, not the white ones, yes. And therefore, they have higher reproduction success, yes. And this is determined by this enzyme. We didn't thought that this enzyme is doing so important function like male choices in birds, yes. There's a third player. We now talked about this BCO2. There's another player, and this is a protein SRB1. That's scavenger receptor B type 1, yes. And this scavenger receptor B type 1 was very early, was characterized in the 1990s as major binding protein for HDL, for high density lipoprotein, and the important role of this protein in cholesterol metabolism was established by work from Monty Krieger's group at MIT. We, in the beginning of the 2000s, yes, we became also interested in this protein because we found a Drosophila mutant, a blind Drosophila mutant, which is deficient for this protein, cannot accumulate carotenoids and becomes blind because of this, yes. This protein is located with two transmembrane, is a transmembrane protein which is located in the cytoplasm membrane. And as I mentioned before, it binds HDL. And what it does, yes, and this was shown, it mediates the uptake of cholesterol, yes. But it was never really clear, yes. And for example, SRB1 is highly expressed in the adrenal glands, yes, and it acquires all the cholesterol for steroid hormones into the cell. But it was never really clear how this cholesterol is now transported to the mitochondria for further metabolism, because carotenoids, cholesterol similar to carotenoid, has to be oxidized. Yes, in this case, they are become hydroxylated, they become heavily modified in steroid hormone synthesis. And then it was Peter Tontonos from University of UCLA, yes, who is neighbor from you, but he nicely showed that a protein class which was not known before, namely the astor proteins, are mediating these transports. Astor proteins, they act like bridges between membranes, yes. They have a membrane binding site on both sides, yes, and a lipid binding site in the middle, and they act like bridges, and they mediate now this transport of lipids, which cannot diffuse in this aqueous environment from one membrane to the other, and finally also the carotenoid, also to the mitochondria. We showed before that SRB1 is not only responsible for cholesterol transport, but also for carotenoid transport. And therefore we saw, yes, it might be logic that these astor proteins are also involved in carotenoid transport, yes. And what we did then was, here first the structure of asters. These proteins are relatively large, 700 amino acids. N-terminus is followed by a gram domain, that's a binding domain for phospholipids. Then you have the astor domain, that's a start-like domain for lipid binding, and then they are anchored in the ER with a transmembrane domain, yes. And here is the structure of astor A, which was determined by Peter Tontonos' group, yes, bound to a cholesterol. We saw, yes, when these proteins are also made in carotenoid transport, we should be able to modulate carotenoid into this binding protease. And I mentioned it hopefully before, that Sepalika Bandara from my lab, she did all this, or most of this work, yes. And so Sepalika modeled it, and we were able to easily modulate a carotenoid in this binding proteins, showing that even this far more rigid molecule fits in this binding pocket. Okay, that's nice work in silico. This does not tell us so much. We can do a lot of things in silico. We really wanted to have biochemical evidence for this. And what we did then was we used E. coli. We established an expression system for membrane extraction of carotenoids by astor proteins. What we did, we used an E. coli strain, which we have constructed before already, which is synthesizing zeaxanthin, yes. And zeaxanthin synthesis takes place in mitochondria via phytoin, lycopene, beta-carotene, zeaxanthin. Here are the known enzymes, yes. And when you're expressing, you can use a plasmid. This plasmid is taken from a pathogenic plant bacterium, which is producing carotenoids. We can produce nicely carotenoids in E. coli. What we then did, we expressed the lipid binding domain from astor proteins now in the system. And what we were expecting is, if this protein is really able to extract carotenoids from mana, we should be able to purify yellow protein carotenoid complexes. And this was exactly what Sipalica did. Here for asta A, here for asta B, here's a control, maltose binding proteins, there are maltose binding protein fusion. You can see, I think with the naked eyes, this golden color, but spectroscopy showed us, yes, that these proteins, yes, now really had very characteristic spectra, which comes from carotenoid binding. What we also found here is that there are spectral shifts in these carotenoids when you compare it to the native zeaxanthin. So this indicated significant interactions between the binding site and the carotenoid ligand. We then also went and tried to do this not only in an E. coli strain, we wanted to show really binding in the test tube. And for this, Sipalica purified asta A protein, here's maltose binding protein, and incubated this now in Triton solution. And we did not use carotenoids because we had problems to bind carotenoids, but what was working really well was apocarotenoids, yes. And here is the spectra shown. And here you can see the binding curve for this apocarotenoids. You have classical saturating binding. And we could also show here in this competition and assay, here we have this dose response assays, yes, binding curves. And what we can nicely see here is asta A binding to cholesterol, NBD fluorescent cholesterol shown here. And when we are now doing this in the presence of carotenoids, yes, we have a right shift, which indicates competitive binding. And here for this, I want to mention, this also is confirming the interaction between the binding sites, because for this we use quenching of tryptophans within the binding region, which are present in this binding region in the astor proteins. And this was quantified by tryptophan quenching assays, further demonstrating there is specific binding. Okay, this is nice. This was test tube E. coli recombinant protein. How is it in cells? Yes, and now what Sipalica now recently did, we searched for cell lines which are expressing high levels of astor protein. And we found one cell line A549. That's a lung cancer cell line, not an ocular derived cell line, but it had fulfilled our criteria. It's expressing high levels of astor protein, and in this case, astor B protein, and it's also expressing BCO2 and other SRB1, other components of the path for carotenoid transport. And so she was incubating these cells in the presence of methyl beta cyclodextrin as a carrier of zeaxanthin. This worked very nicely. And what she could show now is when she is incubating this with zeaxanthin, you get the following result. These cells are nicely accumulating. Zeaxanthin is seen with this yellow color. When we are now looking to the plasma membrane, we do not have so much staining. Here is the cytosol, but here are the mitochondria. It really accumulates this zeaxanthin in the mitochondria of the cells. To demonstrate that this transport is mediated by astor, here is this HPLC for this. To demonstrate that this transport is really mediated by the astor protein, we did siRNA experiments. Here, the control experiment, yes, like before. Here, she used siRNA against astor B, and now compared these two things as here the accumulation of carotenoids, here the downregulation of astor proteins. We were not really able to completely, unfortunately, downregulate astor protein, but as you can see here in comparison to beta-actin, there was a significant downregulation. When we are now separating those cells in different fractions and quantify this, we see we have an increase of zeaxanthin in the siRNA treated cells and a decrease of zeaxanthin accumulation in the mitochondria, providing further evidence that astor B is really mediating this transport of carotenoids into mitochondria in a human cell line. We also try to do this now on the next level, mainly in animals, yes, and for this we look now in mice, yes, where astor B protein is expressed, yes. And astor B protein, this was already published by Peter Tonners's group, is highly expressed in the adrenal gland and at much lower levels in other tissues. There's also testes which are expressing this protein relatively highly. And here's comparison between different tissues. Here, mouse adrenal gland. Here, mouse retina. It's not expressed in the mouse retina, but it's highly expressed in the adrenal glands. And astor proteins exist in different splicing variants. Yes. What we then did, we took advantage of BCO2 mice. Yes. And what we did, we used this BCO2 mice. Yes, and then fed them with zeaxanthin. Remember, these mice accumulate each carotenoid what you're feeding them, except beta-carotene, by the way. But it's nicely accumulating zeaxanthin. Yes. And then we looked in different tissue how it's accumulating, yes. And what you can see here now is in the eye with low expression, there's no accumulation, yes. This dotted line indicates the serum level, yes, of the zeaxanthin in this mice, yes. In the eye, there's no accumulation. But when you're now going to the adrenal gland, you see this really high accumulation of serum levels. And when you now isolate this adrenal glands from this mice. Oh, this was too fast, yes. When you now isolate these adrenal glands from this mice, you see this very nice yellow color. And here, these mice were not treated with that. You see how a normal adrenal gland is. This indicated, yes, that also in vivo, yes, astor proteins, yes, cells which express astor proteins are accumulating carotenoids. In this case, similar to cholesterol in this pyruvate, it also tells us that in vivo, yes, because normally the adrenal gland accumulates carotenoids, that carotenoids really also in vivo can compete with cholesterol for astor protein binding and astor protein mediated transport. This brings us now to the human eyes, yes, and we look now for the human photoreceptor, especially cone photoreceptor, because there is high expression or high accumulation of carotenoids, yes. And when we now looked in the human protein atlas, what we found was that indeed, yes, astor B, yes, the protein which is not expressed in the mouse eye, yes, but expressed in the adrenal glands in the mouse, is highly expressed in cone photoreceptors. That's one of the places of its highest expression in all cell lines which were analyzed by single cell sequencing. And when we looked now, yes, in human donor eyes, we really could show this, yes. Here's the expression of astor B by Western blotting, yes. Here on the peripheral retina, here on the central retina, here on the human mouse retina. Astor B, which mediates transport of carotenoids in cell, in E. coli, but also in in human cell lines, and also in mice, yes, is highly expressed in the human retina. Interestingly, we also found that BCO2 gene is highly expressed in the human retina. So we became interested in the spatial expression of these two proteins. Astor protein is helping carotenoids to go to mitochondria, and BCO2 is in mitochondria and catabolizes those carotenoids. And what I want to mention here is that photoreceptors contain quite a bit of mitochondria. So we got human donor eyes, and they were dissected into peripheral retina and central retina. And we went now and determined by RT-PCR the expression of Gram D1B, that's the other name of astor proteins, and we find that astor proteins are high expressed in the peripheral, in the central retina. They are at least higher expressed in the peripheral retina. But there's also expression in the peripheral retina. When we are now looking for BCO2, this is central retina, is not fovea in our case, yes. You see that BCO2 is highly expressed peripherally and lower expressed centrally. This also has been demonstrated by a German group who showed in the human by single cell sequences that one of the most prominent differences between photoreceptors, cone photoreceptors from the periphery and from the fovea is BCO2 expression. In the fovea, those BCO2 enzyme, which is catabolizing carotenoids, is not expressed. And this would indicate, yes, that this macular pigment accumulation, which occurs here in the central part, yes, is mediated by this differential expression pattern. Gram1D will help carotenoids enter cells both in the periphery and in the central retina. But then BCO2 will make the difference. Yes. In the periphery, carotenoids are most likely converted to smaller entities and destroyed by BCO2. In the central retina, they accumulate. And then we went to staining of the retina. And this was done together with Yoshi Manishi from University of Indiana. Yoshi also worked here at Case together when Chris was here with all of us, yes. And Yoshi did this beautiful staining here on red sets, CRAM D1 in the human retina. Here is the fovea, yes. It's expressed throughout the retina, but when we are now looking in the fovea at higher magnitude, you see this very nicely expression in outer segments, but also inner segments of the cone photoreceptors, which are here on the fovea. And I think this brings us some evidence, yes, that this protein, it's not expressed in the mouse retina, which do not accumulate much kernoids, but it's highly expressed in the retina. That these astor proteins are not only that they are not only generally involved in kernoid and cholesterol transport, but that they may play a specific role together with BCO2 in this very distinct distribution pattern of kernoids in the human retina and help kernoids accumulate in the central retina. What will we do in the future? What do we need to do in the future? That's shown here, yes, with astor B protein. Certainly, it's now evident what we that to really nail down and demonstrate that astor B is this protein which is required for kernoid transport. There are patients suffering from astor B mutation. The phenotype of this patient is that they have that's a recessive mutation, yes, and it causes neurodevelopmental disorders with intellectual disability. And certainly it will be very interesting, or it would be very interesting, to look for the fovea of those patients and see how whether kernoids accumulate there. Another proof of concept would be, we have now this BCO2 mice. These BCO2 mice are now used in several laboratories, including Paul Bernstein's lab, as a model, as macular pigment mouse model, yes. And certainly it would be very interesting to overexpress astor B, which is normally not expressed in the mouse eyes, to demonstrate that astor B is really the key for kernoid accumulation in retinal cells. I'm now at the end of the talk, yes, and I want to thank again my people in the lab who are listed here, yes. They all fantastically contributed, in my opinion, to this project. We had wonderful collaboration. Yoshi, as I just showed, with this immunostaining. We have Adrian Wiss from DSM in Switzerland, who is really constantly providing us with kernoids. He was a former member of the Roche team who synthesized all these kernoids. We have a collaboration with Eckhard Jankamsky here at Case with the deep sequencing analysis. We are collaborating since ever with Marcin Golczak on the biochemistry of kernoids, with Beata on Rhodopsin, and certainly, yes, I'm collaborating with my dear friend Philip on many aspects of kernoid oxygenase. I didn't really have time today to show much about those work, but it's Philip is doing a fantastic job with these enzymes, and much what we know, yes, about these enzymes, including RP65, BCO2, and BCO1, stems from Philip's work, yes. I want to thank the National Eye Institute for their generous support, and thank you for your attention, and would be happy to take any questions. Thank you.
Thank you, Johannes, for a really wonderful seminar. Lots of really good stuff there. So we've got quite a few questions, and I'll kind of just go through these in order of which I see them. So maybe we can start with Dr. Kethalov.
Hey, Johannes, that was a beautiful presentation. Really, really interesting and thought-provoking. A couple of related questions about the STRA6. I was really intrigued by this gradual accumulation of retinoids that you see in the STRA6 knockout mice. Do you have any idea of what is the mechanism that enables this gradual accumulation? I assume this is done in cyclic light, so it's not just the accumulation in the dark. And a related question is, have you thought of, again, you showed that you can modulate, that cone survival also is affected. Have you thought of maybe reducing rhodopsin expression as a way of shifting the balance and allowing more chromophore to end up in the cones and maybe run, sustain their function? Obviously, by using the rhodopsin knockout.
Thanks for the question. I mean, the second question is great and easy to answer. Yes, certainly we would like to have NRL mice cross STRA6 and NRL mice make a cone only model, yes, because it could be, yes, when we are thinking about evolution, yes, it could be that STRA6 is really an invention of vertebrates and is an invention which is very much related to rod photoreceptor vision, yes, because this STRA6 mice are this high demand of retinoids. Yes, it certainly came with this developments of rod cells and nocturnal vision. Yes, with regulation of this, we do not really have a clue. I can tell you a few observations that we had, yes, which is telling you that there is we do not really understand it now, and it's in somehow more complicated. It has more layers than we saw before. Yes, we tried to do vitamin A supplementation, high dose vitamin A. We flooded those mice with vitamin A, and then we thought, okay, when we are giving so much vitamin A, this will end up in vision. Yes, that was not the case. Yes, we do not even find really retinal ester accumulation in the eyes of these mice. In the wild-type mice, we can increase the retinal esters by a lot with this accumulation, but not in the STRA6 mice. So there is more coupling. Yes, either this in and out is more coupled with STRA6. We do not really know why. What we also observed was, and we presented this here at the AVO, Jean was presented this at AVO, that we can rescue the cone vision with beta-carotene, with high dose of beta-carotene. And then the STRA6 mice are doing much better. This simply tells us there is more to learn about these pathways. There is not only this very simple in and out by STRA6 and then it's somehow distributed. There's regulation in this distribution. And that's certainly something what we would really love to figure out, or where we're also looking forward to get that hopefully other groups are working on this and provide input to it.
Okay, quick follow-up. So did you see any upregulation of CRLBP or IRBP or any of the other chromophore binding proteins in the STRA6 knockout mutation?
No, we did not look for CRLBP. So therefore I cannot answer this, but this might be really something what is interesting. Yes.
Thank you. Thanks again.
Hey, Dr. Redmond.
Hi, great talk. And it was great seeing you at ARVO in Denver last week. I had a question that was sort of a follow-on to Vladimir's question, and sort of a comment embedded in a question. But a long time ago, we did a study with David Pepperberg's lab on transport of retinoids in RP65 knockout mice. And I think it might be worthwhile for you to say go back to that paper. It's in IVS in 2003, I think, where there's more than just STRA6. I think is contributing to the to the pool and the stabilization of the pool. And I would suggest that RP65 itself and also LRAT are involved, because what we found was that in the RP65, the wild type, that there was apparent efflux of vitamin A from the RPE, but that this stopped in the absence of RP65, and the retina seemed to, or the RPE seemed to accumulate way more retinoids than in the wild type state, but obviously can't do anything with it. And I think you would lay the blame at the foot of LRAT, because STRA6 cannot export retinal esters, and these just basically accumulated. So I'd be interested in your comments, and I'd suggest that you kind of follow up on this.
That's wonderful suggestion. There was a lot in this literature. Yes, I know this. Yes, I read this. I'm trying to sort it. Yes, and to combine it. Yes, and certainly it will be. There must. This is also based on your work. Yes, there must be communication between these enzymes. There must be something which is telling the system stop here. Yes. Because otherwise we would get this flooding of the eyes with retinoids. we have very specific uptake system. I perfectly agree with you that RPE65 and LRAD are components to which we should look at, yes. And I'm always waiting that somebody comes up with some good idea how calcium may play a role there, yes. And how, for example, this crosstalk between the flow in the visual cycle, yes, and STRA6 is occurring, yes. And I agree with you that STRA6 is certainly a key component. RP65 is certainly a key component in this. And I have to read this work. I wrote it down, yes. I remember this paper, by the way.
Wonderful. Okay. We have a few questions in the room, and then I'll leave the last question to Dr. Okay, maybe Roxana first, and then the last question to Dr. Bernstein. Zach, do you want to start?
Sure. Hi, Dr. Von Lintig. Thank you so much for the really amazing talk. It was very enlightening. So I had two quick questions. One is, does endoplasmic reticulum localization in the RPE play a role in efficient handoff of vitamin A from STRA6 to LRAD and RPE65? So I'm talking more about like spatial localization of the ER within RPE cells. And then the second question was, do you think that there are kind of alternative import or export pathways that get activated with the leaky vasculature? You know, perhaps cells in the retina are scavenging vitamin A that's freely circulating in the bloodstream.
These are very good questions, and the first one I would answer like we do not know. Yes, that's certainly something what we should do. Yes, we have this. We know that STRA6 most likely feeds vitamin A into the plasma membrane. Yes, and we know that CRBP may play a role and to transport it to LRAD. Yes. But we do not really know what is time limiting there and how this is regulated. This is certainly something where we have to look into, yes, in the future. With the leakage, in the, I think, yes, with vitamin A deficiency, we are just starting now to analyze this mice, because I think this STRA6 mouse is simply, I thought it's quite enlightening in such far that before we could not really make a mouse vitamin A deficient. Yes, we know this problem exists in humans, but rodents are very, very resistant. Rats not so much, but mice are very resistant to vitamin A deficiency. Therefore, our idea was simply to use this mouse model as a model for vitamin A deficiency. And one of the major findings is now that it also affects the outer blood retinal barrier. You find those things also in diabetes. Yes, you find this in macular degeneration. And I think, yes, it might be very interesting to analyze what is now really happening there. Yes, and we could probably use this mouse model. I do not know whether it's worthwhile. Yes, and but it could be something where we can learn about this breakdown of this outer blood retinal barrier, what is happening there. And we are just at the beginning to do this. Yes, but good, wonderful questions.
Dominique.
Hi, Johannes. Good to see you. Fantastic talk. This is Dominic from Cristobal. Johannes, quick questions. First, did you guys check LRAD by on transcriptome level, or also by Western blot, for example, protein level in STRA6 knockouts? And the second question, did you compare maybe transcriptomes of STRA6 knockout that were on vitamin A sufficient and deficient diet, to see what kind of genes from retinoid metabolism are changed, upregulated? What could be the other gene responsible for retinoid transport?
This is a very good question. I think we looked. I have to ask why Ram may have done. I think he did. We measured LRAD. We did not see so much differences. Yes, we also looked for the genes of retinoid metabolism in our expression-wise. Yes. On the expression level, there's not so much changes. The only thing that popped a little bit out was RDH5. But with LRAD RP65, let's say with RP65, it's anyway expressed at a relatively high level, yes. So we would not expect, but there is not so much a drastic change. I have to look into this data. We will do this, but I do not really know whether this is on the transcriptional level or whether we really now have to go and look on the protein level, what is going on there. Transcription might be not the answer to this. It might be rather really how this activity of this protein is regulated on the protein level, in my opinion. But we will see, yes. We will publish this, and then you can certainly all look at this data. But it's great seeing you, Dominic.
Emma?
Hi, Dr. Von Lintig. I'm Emma, Dr. Kern postdoc fellow. Thank you so much for answering all my emails all the time. I have just one question. Do you have any hypothesis of why the rhodopsin coupling recovers in the STRAT6 vitamin A deficient mouse versus the cone opsin coupling?
Why the rods recover? I know we do not really have an answer. We saw this, and also others are here in this audience saw this, yes. When you are going with RP65 chromophore deficient mice, in mice it's at least the case, yes. In mice, and somehow the rods steal from the cones when we have too little vitamin chromophore. That's something what is not only observed here now again in this STRA6 study, it was observed with RP65, especially when you're looking for not RP65 null mutations, but when you're going, for example, to the mutation which was done by the Swiss group, yes, R91W, yes, you can show that there is a stealing going on between rods and cones. Why
is that stealing? You know, do you think it's a chaperone protein or another mechanism of transport in rods that is not existent?
I can only answer. With zebrafish, we have some evidence, yes, that CRLBP plays an important role there, yes. That this is helping cones to get vitamin A. We have now this observation, for example, that beta-carotene can rescue cones, yes, in STRA6 mice, and somehow when it's genetic manipulation, it's not straightforward. So there is evidence for other pathways, but it's a very pressing, important question, but I can't answer it. I'm sorry.
Thank you.
Aisha?
Hi, Johannes. This is Aisha from Dr. Kern. Very nice talk. One of my questions was taken by Emma, but I have the second question. I am interested in understanding what is going on in terms of calcium in the STRA6 knockout. I know that you said there is a relationship between calcium homeostasis and STRA6 enzyme.
This is not work by, that's a fantastic question. It's not work by all. There's work from Hui-san's group who showed that the opening and closing of the channel is dependent on calcium, yes. But we do not, what we do not know now, yes, or currently, or at least we don't know, probably others know, yes, it's unpublished, but how the visual cycle and this opening and closing via calcium, whether there is really a connection, yes. And that's something where this is something what is really, in my opinion, very important to do, yes. And there are calcium reporter cells and a lot of things what you could do, yes, in the future. But I think it's very worthwhile to investigate this because we do not know how this is regulated and how this homeostatic pool is maintained.
Great. Dr. Radu.
Hi. Johannes, this was excellent work presented today. I'm interested if you can speculate about the crosstalk or, somehow role of the non-visual opsin in the RPE in relationship to STRA6 modulating the vitamin A. At least two of them has been shown to really play significant role in homeostatic of vitamin A, of course, by mechanisms that I'm not sure are clearly understood. For peropsin, for example, dark adapted state, the RP has significant lower level of the storage level of all translated palmitate. However, in light, those knockout mice seems to recover the level of vitamin A to the wild type level. And for RGR, obviously it shows that mobilize the retinal astor in light. So clearly has a, sort of light mediated role. And how would this, kind of affect the dynamic of STRA6?
Again, that's a wonderful question. I'm sorry that we looked at it, let's say with beta-carotene. The only thing what we did with beta-carotene, because we had certainly this work about peropsin and RGR on our list, yes, because we are interested. And we do not have the knockout mice, what might be. We are happy to share our STRA6 mice with whoever wants it. And but they are available in several groups. But what we did with beta-carotene, because we had this beta-carotene rescue for somehow, so we thought, okay, beta-carotene, BCO1, which is not very highly expressed in the mouse eye, but there's a little bit of expression, may feed to RGR. So we did this rescue light and light independently because we would expect when it's RGR dependent, it should not occur in darkness, but this was not the case.
So in STAR6 mice, the expression level of those non-visual opsins are changed?
No, we did not. We did not look to them. We will. It's I'm getting a lot of good ideas today from you delivered, yes, and good future directions. I'm really happy that I had the chance to present here. Thanks so much. But I'm sorry, I cannot really. I have no answer to your question. I only can mention that the beta-carotene rescue of cone vision, what we observe, yes, is not light dependent.
Thank you.
Okay, so we're getting close to 9:50, so I think I'll stop with questions now and leave the final word to Dr. Bernstein.
Okay, oh, thank you. I have, very good to see you, Johannes. I have comments and questions on each half of your talk. The first one with regard to your mouse models of vitamin A deficiency. Clinically, as a clinician, I see patients with vitamin A deficiency, and it's very clear that they lose rod function much earlier. Cone function is preserved all the way to the most deficient patients. I'd just like for you to comment on about why this disconnect may be occurring, if you have any ideas.
That's a great question, Paul. Let's say it like this. In mouse, you always observe this thing. Yes, with you have the same with RP65. You cannot compare it. Yes, I will answer like this. Yes, with RP65, you also see in humans differences to mice. Yes, because you have a loss of cone vision. Also when you are going for this less severe variants. Yes, you have a absolute loss of cone vision, whereas rod vision in somehow can be a little bit maintained. What is also the opposite to humans. Yes, I will not say that this mouse model is the last answer to vitamin A deficiency. It's more an answer where it's more a model because normally you cannot do this in mice. Now we can do it relatively selectively, and we do not affect the rest of the body, what is also good. Yes, because with the other mouse models, mice when they are really becoming vitamin A deficient, that they get visual problems, they die. Yes. And this is not the case here. That there are differences between human and mice, there is no doubt about this. I think that the distribution of vitamin A into cone and rod pathways, there is differences between mice and humans. That's my experience. And I think we have to either go to other models. Yes, zebrafish might be a little bit a solution because there you can see that there are different solutions to this. Yes, and I think this is also represented when we are looking for the retinoid biochemistry, yes, and I do not have to tell you this. I'm one of the first experts in this. It's like this, for example, we have this accumulation of 11 cis retinal esters in the human retina, not so much in the mouse, not retina, also in the RPE, yes. And I think there are significant differences between nocturnal and diurnal animals, yes, and this may explain at least some of these differences. That these things that what I presented today doesn't answer everything. That I have no doubt about this. It's only something where we can build on. Yes, we will see. Yes. Also, what I presented about carotenoid binding transporters, that's not the only thing. What we are having here, that's a new player. We have other players. We have now to figure out how this works together.
Yeah. So my second question follows on just to what you said. As you know, we've looked at other binding proteins, including STAR-D3 for lutein and GSTP1 for zeaxanthin. And it's nice to see another player in this. I really think that's great work. The one thing that's an interesting disconnect is in my work on MACTEL and others who are working on MACTEL, it seems to be that we know that carotenoid concentration is disrupted in the macula, but it appears from a lot of their work that appears to be in the Mueller cells more than anything else. And I think your work today and our work shows that most of these binding and transport proteins localize to the photoreceptors instead. So I don't have an answer to that. I don't know, and you may not either, but it's an interesting finding. I'd be interested in your comments on that.
your work on Martel too, yes. I also looked very much in, I read all this because I'm very interested. What I found very interesting, at least when you are going for this one patient whom you analyzed, where you had the retinal, yes, that you saw this mitochondrial degradation, yes, and I thought that's very interesting, yes, and this may tell us that there is a connection between carotenoids and mitochondria in the human retina, yes. But there's a lot of things. There's a lot of things which is left to do. Yes, and it's the same with this asta proteins, because the asta proteins localize to the outer segments, yes, and also to the outer segments where no carotenoids are. Yes, and what the asta proteins is, these are certainly this is bridges for carotenoids, so they can enter the cells. Whereas the carotenoids then finally accumulate now, they are then handled there. Yes. That's there are other players may be involved, yes, and it's we have more players now, and we have more to do now. What is always good, I think so.
Yeah, very nice work.
Wonderful. Well, that was a super great discussion. Thank you, Johannes, again for presenting, and all the attendees for the great questions. It's fantastic, and thank you so much, and we'll see you all next week.